Thanks again for the organizing committee for inviting me to present our research on powdery mildew genetic resistance in this conference. It's a pleasure for me. I'm lead pathology scientist at Aurora Cannabis Company, located in Canada. Our facility is a dedicated breeding and genetics facility. We're focused on different breeding projects from aroma. It's one of the projects going on. Plant architecture, potency, and disease resistance. This is the project that my team and I are focused on. Whenever I had this experience that when I introduce myself as a plant pathologist, particularly with the people who do not have plant science background, I'm getting this question that, oh, really, the plants have immune system? And so it doesn't look like that they have this complex immune system. So the answer is, of course, plants have been around for at least 500 million years. That's the evolution of higher plants. And they're still around. So they've been able to defend themselves against all these microorganisms, fungal, viral, and bacterial pathogens that are trying to infect these plants. They do have an immune system, and they have an adaptive and inducible immune system as well. It's pretty sophisticated in its own kind. However, it's not as complex as our mammalian immune system. Plants do have a multi-layered immune system, and I have a pointer, so I'm the first speaker today that can use this. Thanks, Alex. This metaphoric onion showing the multi-layer nature of plant immune system. At the outer layer of plant immune system, we have preformed defenses like cuticle or trichomes. Secondary metabolites of preformed antimicrobial compounds. And we know that like THC and CBD, they have some antimicrobial properties. However, in the heart of the plant immune system, we have an inducible defense mechanism or immune response, where the plant host is able to precisely perceive the presence of the attacking pathogen and adapt and tailor its defense response according to the nature of the attacking pathogen or invading or penetrating microorganism into its tissue. Plants do have certain receptors that can sense the presence of a specific pathogen, and then after the perception of the presence of the pathogen, there are upstream signaling molecules, nitric oxide and hydrogen peroxide are among them, that start a signaling cascade and transduction pathway. Some hormones play a role in this signal transduction. We had a speaker previously that spoke about salicylic acid, jasmonic acid, ethylene. These are important defense-related hormones in plants to transfer the danger signal to the nucleus, where defense genes are located. And these are the genes that can, based on the type and nature of the pathogenic attack, can define how the plant is going to respond to that pathogen. There are several different genes, genes that produce specific antimicrobial compounds, proteins called pathogenesis-related proteins or PR proteins, and a programmed cell death response, which is specific to plants. It's called hypersensitive response. We'll be seeing the nature of that response in next slides. And this is a type of response that plants can fortify its cell walls to prevent the attacking pathogen from penetrating into its tissue, accompanied by the accumulation of several antimicrobial compounds. So I just told you that plants are pretty responsive to their environment. They can sense the presence of a pathogenic attack, but how responsive it is. There's a video here. It's fluorescence imaging. That's why you don't see that much right now. It's top view of an Arabidopsis plant. It's a great work done by Dr. Toyota from Saitama University in Japan. what they did, they added a molecule here, a compound here that can mimic pathogenic attack, particularly wound response in plant, on this side of the plant. And they had this GFP, green fluorescence protein, conjugated to a signaling molecule. It's called calmodulin, which is triggered by calcium ion. Long story short, when you see the fluorescence illumination throughout this plant, that's actually an indication of the danger signal being spread through the whole plant tissue. So just imagine that's the initiation of the observation. We have this danger signal molecule put on the plant, and then the video starts. I cannot click on that play, so Alex, if you could please play that video for me. Thank you. So you see, molecule is here, and boom. The whole plant is right now alerted about the presence of that pathogenic attack here. It's 298 seconds, but if you stop the video at around 80 seconds, that's where this part of the plant is showing response to the attack on the other side of the plant. So 80 seconds for the whole plant to get that danger signal. So plants are that responsive to their environment. Next time you're pruning your cannabis plant, just remember that it knows that you're around. Don't want to go into details here, but that's adaptive or inducible plant immune system. Because if you probably know, and you saw it in the first slide, we just discovered and identified a new resistant gene in cannabis plant, which is effective against powdery mildew disease. And the question here is that what is a resistant gene? Resistant genes are a specific classification they encode a specific classification group of proteins. And these are the proteins that can perceive the presence of pathogen. So there's no relation to the final outcome or downstream of plant defense mechanism. It's just at the upstream where the plant is able to perceive the pathogen. There are multiple defense responses that can be activated after that interaction between plant receptors, which are R proteins or resistant genes, controlled by resistant genes, and a pathogen-derived protein. So pathogens, when attacking the plant, this is pathogen-triggered immunity. They release some molecules. There are broken down molecules coming from the cell wall of the pathogen, for example, chitin from fungal pathogens, that can be perceived by broad spectrum receptor-like kinases, transmembrane protein, which initiate a broad spectrum defense response. Evolutionarily speaking, this arms race or ping-pong-like interaction, evolutionary or co-evolutionary interaction between plant and pathogens, pathogens have been able to kind of suppress that mechanism by injecting certain protein molecules called effectors into the cytoplasm of plant, that these molecules can suppress that broad spectrum, it's called PAM-triggered immunity in plants. Resistant genes encode proteins that can perceive these specific proteins injected by pathogen into the plant cell. So these are the proteins and genes that we are after. If we can find these genes and some genetic resources available to us, then we can use those genes to make new cultivars or genotypes of cannabis plant or any plant and make them naturally resistant to that pathogen and disease. What is powdery mildew? I'm pretty sure that you have heard the name powdery mildew. Some of you, I know that you're specialists on the matter as well. Powdery mildew is a fungal disease that is right now one of the most economically important diseases of cannabis, which threatens the cannabis industry. There is 3 important stages of this disease, fungal disease. First stage, we have the spore of the pathogen, and this pathogen is mainly airborne. So spores are in the air, landing on a new leaf, and then it's germinated, and they penetrate into the tissue of the plant. They use high level of pressure in an organ called aprosorium to punch or drill, let's say, the cuticle and cell wall of the plant. Some of the powdery mildew species have pressure twice the pressure of your car, a tire car, about 8 pascals, 8 bars, which is amazing. So it's highly melanized organ that can produce that amount of pressure. It can easily punch an aluminum foil. So that's the amount of pressure. Sometimes I get this comment that if we have some new genotypes with thicker cuticle, do you think that that might be resistant to powdery mildew? The answer is obviously no. After that penetration, the pathogen is able to make this organ. It's called hostorium. And you should consider that powdery mildew is a parasite. It needs the cannabis plant or any other crop to be alive. It does not kill your plant. It keeps it alive, and it drives nutrients still the host is alive. And this organ does that job. It makes an cytoplasmic interaction with the plant cell, and it drives all nutrients from that plant cell. And it suppresses plant immune system in the plant cell at the same time. It injects several effector proteins that I just introduced to you to do so. The second stage is that where the pathogen develops its mycelial network on the surface of leaf and then makes these vertical structures for its reproduction. This is called conidiophores. And these are actually the last stage of the pathogen lifestyle where it produces millions and millions of new spores. And these spores are ready to fly in the air and go to a new plant and then just redo the life cycle of the plant. This is a very interesting image. This is not a photoshopped Halloween image. That's electron microscopy done by my friend and colleague, Marco Tedesco. It's on Arabidopsis. So you can see, as I told you, the mycelial network already developed on the plant. And then these are the vertical structures coming up, making the new spores. And that's actually the completion of the life cycle of this pathogen. Okay, the economic impact of powdery mildew disease. As you know, it's been shown that even mild level of disease occurrence at LPs, licensed producers, it can cause losses up to 30% of the yield. And to control this disease, although it doesn't kill your plant, it's a biotrophic pathogen, as I told you, you need to apply several IPM products, integrated pest management products, which are expensive, and it's labor intensive. You need to have staff to apply those products, doing regular scouting to monitor your growth zones. There are a lot of activities which are, again, labor intensive and expensive for any cannabis producer. So developing new genotypes of cannabis which are resistant to powdery mildew disease obviously makes a lot of sense. We collected a diversity panel, a diversity population of different cannabis genotypes from different places, like about 550 genotypes. And among them, 108 genotypes were land races and exotic genotypes. These are genotypes that coming from mountains in Afghanistan, from China. We had genotypes from Syria, Middle East, Colombia. I personally chose part of those genotypes. I wanted to have genotypes that are, evolutionarily speaking, they're growing in the areas with high level of humidity. My understanding was that probably these ones are a good source for genetic resistance. You know, the problem with available germplasm of cannabis is that because of the years of cannabis production being illegal, the black market people who grew cannabis, they didn't care about genetic resistance to diseases. Their selection was mainly based on potency or flower formation, all those agronomic traits. And they applied a lot of fungicides to control diseases. So the genetic pool, available genetic pool in the market, unfortunately lack resistant genes or R genes that are effective against important diseases. That's why we have to construct new populations comprising of land races and exotic races to be able to detect if there is any R gene to detect those R genes. And that's what exactly we did, and you can see these wonky plants growing in our facility are mainly land races. This is a statistical analysis of the population that we were working on. It's called principal component analysis. It's a way to know the important factors on your population and which data set and factor that you're using actually forming the structure of population. It's actually dimensionality reduction method in the statistics. I don't want to go into details here, but something really important, and this is the first time that we did, anyone that did this analysis on cannabis plant as far as I know, the question was, what if we find a resistant gene in a landrace, and we use that landrace to breed new genotypes of cannabis, but that resistant gene comes with a lot of unfavorable, that might happen, unfavorable agronomic traits, because these are landraces, they're not good sources of or genotypes agronomically for any elite cultivar. So here you can see we tested several different traits here. The length of the plant, length of branches, leaf formation, potency level, because it's believed that THC, CBD have antimicrobial compounds, so some of this, it's another thing that I need to say here, that a lot of pseudoscience, as you know, that are available online and about cannabis plant, so some people call it bro science, that believe that higher potency means better disease resistance in cannabis. At least for powdery mildew, that's not the case. Or some believe that cannabis indica, and I know that this right now, indica sativa story is already resolved and it's not supported by genetic studies, but it's believed that those indica leaves are more susceptible to powdery mildew and those sativa leaves are more resistant. So we wanted to know how is the situation here. And interestingly enough, so we tested our population with different factors affecting the structure of a population, and you can see that disease index is not correlated with any of these agronomic traits that I just mentioned. And that's the numbers, probably it's more readable, that you can see there are different correlation with different agronomic traits, but disease index remain totally unrelated to any agronomic trait. This is good news, because that means that we can breed for this powdery mildew resistance while still having high level of potency, while still have a, in terms of plant architecture, a favorable architecture of the plant. If dwarfness is favorable, we can do that. Or flower formation or harvest index is not going to be, at least based on the population that we test, is not going to be affected. And that's a population that we use for our breeding purpose. This kind of studies are called GWAS, so genome-wide association studies. It's a known way of detecting new sources of disease-resistant and other agronomic traits in breeding studies. So that's how our population responded to powdery mildew infection. And you see that's this histogram showing a left-skewed distribution, meaning that majority of genotypes that we tested were highly infected, susceptible to powdery mildew, and we had few genotypes that showed high level of resistance, and here it's the disease index. So 0 means highly resistant, 100% is highly susceptible with no any defense response to the pathogen. And unfortunately, or fortunately, I don't know, I had to redact these genotypes names and because of the IP concerns that Aurora company has. Anyway, so there are few genotypes that we detected that showed high level of resistance, so there is hope. My presentation right now is more on the pathology side of the story, but for the breeding purposes, one method to transfer these genes, because this is another question that I get, that is it GMO, so how do you transfer these resistant genes to elite cultivars? No, this is being done through classical breeding approaches. One method is backcrossing, and I will be available after my talk if you have questions about that part. But by doing multiple crossing of our donor genotype with elite cultivar, we are able to transfer that piece of chromosome that contains our resistant genes to our elite cultivar, and making the proportion of elite cultivar, genome of elite cultivar, higher and higher until 99% of the elite cultivar and the resistant gene, the final product. I want to share here a couple of interesting defense mechanisms against powdery mildew that we observed in our GWAS study. This is the first one. So you see the plant on the left is totally resistant to the pathogen. It's a landrace here, and that's a susceptibility situation. And this is hypersensitive response that I just introduced to you. It's a type of programmed cell death that the plant perceives the presence of the pathogen. It kills its own cells in order to make penetration impossible for the pathogen. And by doing that, it also fortifies its cell walls. So you see those patches that are, you see the chlorotic patches here and there. These are where programmed cell death and hypersensitive response is going on. These are where, if you put it under microscope, you will see this. All these purple dots, it's a specific stain that we use that can detect, I'm almost done, it's just 2 more slides. Sorry for that. A specific dye that can detect lignification and cell wall fortification. And you can see just like an ancient fortress where you need to keep the walls and keep the enemy outside of the gate. This plant is able to keep the pathogen outside of the gate and castle. Another interesting resistant response, it is not through hypersensitive response, totally different defense mechanism. It just destroys the mycelial growth of the plant. So apparently some antimicrobial compounds are being secreted, and you can see there is no mycelial growth on this plant. Very interesting. And that's the immune system that we saw. And by immune system in plant pathology, we mean that you don't see any reaction in your plant whatsoever, as if there's no pathogens. No difference. You cannot see anything. And under microscope, you see this plant has both hypersensitive response. These are the fortified cell walls, these specific cells. That is where the pathogen is trying to penetrate into the tissue. And then also, we don't see any conidiophores. The pathogen is totally unable to produce any spores. And okay, skip that. This is the final slide of the presentation. We finally found a very good candidate for our breeding purposes. We just filed our patent 2 weeks ago. This is a gene, single dominant Mendelian gene, which means that by any cross that we make, the progenies will have the resistance. So that is highly favorable for breeding purposes. There's a long list of characteristics that you need to have for these resistant genes. One of them is to be single dominant. And you can see that in a susceptible situation, you have these spores being generated, conidiophores, but on the resistant one, PM2 gene, because PM1 has been published, if you follow this story, by Oregon CBD a year ago, and this is the second gene that being discovered here, second locus actually, that is called PM2 now, and you see that the plant remains totally free of the disease, of the pathogen, and the pathogen is not able to produce any spores. So the reproduction phase of the pathogen is fully suppressed by this single gene. Thank you so much for your attention.
There's time for one question. Great talk, by the way. I love the topic. Have you tried looking at this marker in other cultivars to see if it still holds true in other lines?
Yeah, we have made crosses with our elite cultivars. Its backcrossing is going on, and yeah, all of them show the high level of resistance. Thank you so much. Oh yeah, another question, please.
Yeah, sorry. Great work on the mildew, by the way. I wanted to ask you if the defense response, does it alter or modifies the number of cannabinoids? Did you do a test?
No.
So, yeah.
We didn't find any correlation with the level of cannabinoids and defense response.
Okay, thank you.
You're very welcome.
Please, yeah, last one. Is the mutation PM2 knockout or just a change in amino acid? Do you know? Can you tell?
The PM2?
Yeah.
I know that it's after finalization of patenting, that will be used internally to develop new cultivars. I mean, it will be added to the elite cultivars because our facility has a business side, is OCO Innovation. You want to check it, OCCO. So that's the plan for this gene.
Okay, but is the mutation leading to a knockout of a gene, or is it just a change in amino acid, changing the sequence, or?
The activity of PM2?
Yeah, the mutation.
The mutation. So what kind of mutation PM2 might have? I'm available right now after the talk, sorry.